El día de hoy tenemos una conferencia muy especial. Tenemos el gusto de tener entre nosotros al doctor Steven HEBERT de la Universidad de Yale. Él es el jefe del Departamento de Fisiología y Bioquímica Molecular de la Universidad de Yale. Yo conocí a Steve hace 17 años cuando tuve el privilegio de ser estudiante en su laboratorio. Trabajamos durante varios años en el Hospital Brigham and Women's y desde entonces hemos mantenido una colaboración que ha sido muy exitosa y que sigue siendo hasta el momento. Steve es médico por la Universidad de Florida y después realizó su residencia de medicina interna y de nefrología en la Universidad de Alabama, posterior a lo cual se mudó junto con su profesor Tom Andrioli a la Universidad de Houston, donde inició su carrera como investigador. Dedicó los primeros 10 años, y esto es durante los 80s, a describir los mecanismos de transporte de sal y de potasio en la nefrona, particularmente en el haz ascendente de Henle. Es a él a quien debemos la descripción original de los mecanismos de transporte en este sitio. Y después, cuando yo llegué a su laboratorio, dedicamos los siguientes años a la identificación molecular de estas proteínas. Steve ha sido muy exitoso en este campo, logró llegar, lo que poca gente hace, logró llegar a full professor en la Universidad de Harvard y posterior a eso se fue de ahí. Estuvo 3 años en la Universidad de Vanderbilt como director de la división renal de esa universidad y posteriormente fue atraído por la Universidad de Yale para dedicarse a la fisiología molecular en plan. Él ha sido muy exitoso en 3 diferentes áreas conectadas pero distintas a la vez. Una de ellas es el transporte de sal a nivel renal, la otra es el transporte de potasio, que es probablemente su molécula favorita, y un área que surgió cuando yo trabajaba con él, y fue el hecho de que él junto con Ed Brown concibieron la idea de que la forma en que el calcio podía regular la secreción de la hormona paratiroidea era a través de la existencia de un receptor de membrana que pudiera sensar el calcio y traducir los mensajes como cualquier hormona, y con esto poner al calcio en papel de hormona. Esta idea, que originalmente no fue muy bien recibida por el mundo endocrinológico, finalmente resultó ser así con la identificación molecular de este sensor, y pues a partir de ahí el sensor de calcio ha mostrado no solamente ser importante en la endocrinología y en la nefrología, sino en múltiples áreas. Y lo que vamos a escuchar hoy es algo muy novedoso en relación a la importancia del sensor de calcio en el área gastrointestinal, que son trabajos que Steve ha hecho en los últimos 5 años y que ya verán que son realmente interesantes. Steve, thank you very much for coming.
Thank you, Corrado. Good morning, everyone. It's a pleasure to be here again. I think this is my 4th or 5th visit to Mexico at the invitation of Corrado and others. And I want to present something a bit different than I usually present. It's not something that has to do with kidney or kidney disease specifically. And it's looking at a problem, acute diarrheal illness, and I think everyone in the audience knows a lot about this. If you look at infectious diseases worldwide, this is data from 2000. About 10.5 million children less than 5 years of age will die worldwide of infectious diseases. If we look at the causes, 7 out of every 10 of the deaths are attributable to 5 main causes: pneumonia, diarrhea, measles, malaria, malnutrition. Diarrhea is the 2nd leading cause of death in children in this group less than 5 years of age. Now, if you look at the number of episodes of diarrhea, and the number is hard to get, the WHO has probably some of the best data, but these are just estimates, and they may even be on the low side, but approximately 4 billion episodes of diarrhea occur worldwide. There's approximately 3 million deaths, although the number you can read anywhere between 2 and 3 million deaths per year. About 80% of those are children, and about 20% are adults, adults meaning more than 15 years of age. It's really a severe and lethal disease. 2 to 3 million children die each year from diarrhea. Somewhere between 500,000 and 1 million adults. And children in many areas of the world will have multiple episodes in a particular year. And in these children, it can lead to worsening malnutrition, slow growth in these infants and children, delayed mental development, increased susceptibility to other infectious diseases, and certainly in the adult population can lead to reduced productivity and quite a severe economic impact in these countries. Well, what causes diarrhea? Well, the problem is that there are many causative agents, from bacteria to viruses. Rotaviruses, certainly an important cause of viral-induced acute diarrheal illness, parasites. And in many of the studies that are done, it's not uncommon to see upwards to 50% of the cases where the individual causative agent is undetermined. Presumably it's some bacteria, parasite, or virus. So the real problem is there are many different toxins produced by bacteria or factors produced by viruses, many disease-causing factors. How do you attack this problem? Well, traditionally, antibiotics have been used. Problems with antibiotics, of course, are development of resistance, or they may have no effect on viruses, or little effect on certain parasites. Vaccines has been also very important in looking at particularly cholera and viruses, most notably recently rotavirus, where 2 rotavirus vaccines have been produced by pharmaceutical companies. And of course this, we're looking at what the impact of this will be. Certainly it can have a significant impact in developed countries. The issue is always delivery of vaccines to the individuals in developing countries, the cost of the vaccine. Vaccines have to be made for, in general, individual viruses or bacteria. So one would have to ultimately look at the development of many, many vaccines. It's a relatively high cost and many times difficult to produce these vaccines. So instead of attacking the germ, try to stop the attack at the patient, that is the host, and that would be the ideal way to attack this problem. It's not curative, it's not particularly preventative, but if you have an individual with acute diarrhea and you can stop that effect at the host, then the potential is that you could save many, many lives while other types of therapy are being developed. Today I'm going to talk about a potential for the use of agents that activate this calcium-sensing G-protein coupled receptor as a potential therapy for acute diarrheal illness. Now, calcium is a very important mineral. It's important for bones. As we know, it's very important as a cofactor for enzymes, both intra and extracellular enzymes. It's an important second messenger. But calcium is also a first messenger. You can think of it as a hormone. And calcium binds to its cognate receptor, which I'll talk about, or sensor, and activates a whole host of cellular responses that are appropriate for the level of calcium. Now Ed Brown and I first cloned the calcium receptor from the parathyroid, from cow parathyroid, and that's because we knew that there was a very steep relationship between plasma ionized calcium and the release of PTH, parathyroid hormone, and this is an inverse relationship, as we all know. So the higher the calcium, the lower the PTH secretion. And since PTH is, parathyroid hormone, is important for liberating calcium from bone, as the calcium becomes lower, there's an increased PTH, an increased liberation of calcium from bone to replenish the plasma pool of calcium. So we used this cow parathyroid to clone the receptor. Many people at the time didn't believe there was such a receptor, and there were some real reasons why one might be skeptical that this was a G protein coupled receptor, and that's really shown on this graph. And that is, you have a G protein coupled receptor with an EC50 in the millimolar range. No such receptor had ever been identified or shown. And despite having this very high EC50 in the millimolar, not the nanomolar range, there was a steep relationship between the change in ionized calcium and the release of PTH. So this certainly was a very, very unusual G protein coupled receptor. Well, we cloned this in 1993, and it clearly was a G protein coupled receptor, and it belonged to class 3 of the G protein coupled receptors that I'll show you in a minute. Well, this was important for parathyroid, regulation of parathyroid function and PTH release. And I think we all know that in renal disease, as renal function begins to deteriorate, there is a hypertrophic and hyperplastic response in parathyroid. And associated with this growth of the parathyroid gland and secondary hyperparathyroidism, there's an increasing loss of calcium sensing receptor expression on the chief cells in the parathyroid. So this led to the notion that if you could activate the receptor with a new kind of agonist, that you might be able to suppress PTH secretion in this hyperparathyroid condition. And the search went on for drugs that could activate this G protein coupled receptor. And the screening revealed a number of aromatic compounds, and one that commercially made it through drug trials is cinacalcet hydrochloride. And this looks nothing like calcium. It's not charged. It's an organic molecule, and it doesn't work by attaching to the extracellular domain of the receptor. It's not a direct agonist, but it's an allosteric modifier. So it works by increasing the transmission of signal from calcium binding to the extracellular domain to the generation of second messenger signals in the cell. It's a lipophilic molecule. It's rapidly absorbed and binds to membrane spanning domains that are within the cell membrane. So it is digested orally, moves very rapidly into the bloodstream, binds, enters cell membranes, and if the receptor is present, it will bind to the membrane spanning domain of the receptor, and it greatly activates the downstream signaling of the receptor. And this has become an important part of the therapy of many patients with end-stage kidney disease and secondary hyperparathyroidism. And this is just one of the studies showing a trial that was done, a phase 2 trial, in which individuals with end-stage disease on dialysis had very high intact parathyroid levels. And they were either given nothing but their routine therapy with phosphate binders and vitamin D, or their routine therapy was continued, plus they were given this calcium medic or calcium receptor activator agent. And you can see over a period of weeks the PTH levels came down, remained relatively stable. There was some up and down, but again remained lower than during this 54-week trial than the placebo controls. And then when the study ended and became off-label, then these individuals were started on calcium medic and their PTH levels came down. And so this is a drug that's been on the market for a few years, and it is used by many physicians to treat secondary hyperparathyroidism. So the calcium sensing receptor, as I indicated earlier, is a member of the class C of the G protein coupled receptor superfamily. So these are many, many hundreds of receptors are in the superfamily. They're divided up into class A, B, C. In class C, you have a group of receptors that are predominantly expressed in the nervous system. And very interestingly, the calcium sensing receptor sits in the group subgroup within class C of pheromone receptors. So last night I talked a bit about the importance of the calcium receptors sitting in this group of pheromone receptors. I'm not going to discuss that today, but I wanted to just indicate that this receptor is acting more like a sensor than a typical receptor and fits very nicely within the kind of thinking that one has about these kinds of pheromone receptors. So the calcium sensing receptor, and this is important for the discussion today, is much more than just a calcium sensor. It's really an extracellular environmental sensor. So what do I mean by that? You can think of the receptor in terms of something like a pheromone receptor. It's a nose sitting on the cell that's sniffing the extracellular environment. Of course, its primary agonists are divalent minerals, calcium, magnesium, but also a very important agonist of the receptor, working predominantly as allosteric modifiers, are amino acids and polyamines. Now, amino acids and polyamines are in everything that we eat. Polyamines are produced by every cell from bacteria and above, so bacteria, plants, animal cells, all make polyamines. So when we eat a meal, we are delivering a load of polyamines. And of course, amino acids are the digestive product of proteins in our meal. In addition to calcium and these nutrients, the receptor is also senses certain factors in the environment. It's highly sensitive to changes in pH and highly sensitive to changes in salinity. And yesterday I talked a bit about the potential importance in evolution of the sensing ability of this receptor for salinity, particularly in the evolution and development of marine species. But this wide group of agonists of this receptor point to the importance of this receptor more than just a calcium sensor and an extracellular environmental sensor. Now, when we began to look at where this receptor was expressed, of course it's expressed in parathyroid cells and in the kidney, but it's expressed in many, many other cells, and including the gastrointestinal system. It's expressed in the stomach, and what I'm showing you in these slides are images from the human, and these are this is gastric tissue that came from individuals who were obese and underwent partial gastrectomy as a treatment for their obesity, morbid obesity. And the red staining is the calcium receptor, and the green staining is the gastric HK-ATPase, or the proton pump in the stomach. And in all the cells that express proton pump, there is expression of the calcium sensing receptor, and it's expressed on the blood side of these cells. And you can see one of the gastric glands here. This is the lumen of the gastric glands, parietal cells around there, and the calcium sensing receptor is expressed on the basal side of these cells. It's also expressed along the entire intestine, small intestine and large intestine. And this is a colonic biopsy from a human. This is a cross-section of a crypt. This is the lumen of the crypt. This would be the blood interstitial side. These are the cells. And the calcium receptor is expressed both on the luminal-facing side and on the blood-facing side of these cells. Now, if you look at rodent tissue, experimental animals, where you can get a better and more detailed look at the expression, this is a colon. Its invagination is a colonic crypt. You can see it here. This is phase, and this is fluorescence. And the red fluorescence is a calcium sensing receptor, and you can see it expressed on the apical portion as well as the basolateral portion of these cells. This is immunohistochemistry, again, of the rat, and you can see the brown coloration both on the luminal and the basolateral sides of these cells along the crypt and along the surface. And you find the same staining in the small bowel, and the calcium receptor in the small and large intestine is expressed both on the gut luminal side and on the blood side of all the cells lining the gastrointestinal tract. So this is from a review that John Goggle and I wrote a few years ago. In thinking about this, the role of the calcium receptor in the gastrointestinal tract and its functioning as a nutrient sensor, not only sensing and responding to mineral nutrients, but also polyamines, as I said, are in everything that we eat, and in amino acids. Now with amino acids, the receptor can be activated by all L amino acids. The most potent amino acids are aromatic amino acids, such as tryptophan. But all amino acids will activate the receptor. Now the expression of the receptor in the stomach, there have been a number of papers published, many from our lab, as well as others. It will stimulate acid secretion if you activate the receptor. And in fact, it's as potent as the major secretagogues like histamine or carbachol. So it's a very potent stimulator of the gastric acid secretion, both directly stimulating acid secretion from parietal cells that have the HK-ATPase, but also from it will stimulate the release of gastrin from G cells. So it's a bimodal aspect of stimulation, very important stimulation of gastric acid secretion. We think this is a feed-forward kind of mechanism, and the idea behind this is that as we eat a meal, we stimulate gastric acid secretion by a number of factors, including mechanical. And this first wave of acid secretion helps to begin the digestive process. This results in the liberation of amino acids. The amino acids are rapidly absorbed, and the amino acids in the bloodstream will potently stimulate acid secretion, and we've shown that physiological concentrations of these amino acids are potent stimulators of gastric acid secretion. This continues gastric acid secretion as long as there are amino acids being liberated from digestion, an important part, we believe, of the digestive process. I'm not going to talk any more about this. This is really an interesting story, but I'm going to focus today on the intestine. And I'll show you the data that it's a major regulator of intestinal fluid transport, and we believe it can be an important player in the regulation or the reduction in fluid secretion and enhancing fluid absorption in toxigenic diarrheas. So if we look at toxigenic diarrheas, cholera toxin can, as an example of a severe diarrhea, that the secretion of fluid can come both through direct stimulation of the epithelial lining of the intestine. We also know, and I'll show you some evidence for that, the calcium sensing receptor is expressed in the enteric nervous system, and expressed in the enteric nervous system both in Meissner's and Auerbach's plexus along the entire intestine, small bowel and large bowel. And the second type of stimulation that can occur in acute diarrheal illness is via stimulation of the enteric nervous system to release vasoactive substances like vasoactive intestinal peptide, which results in enhanced secretion of fluid in the intestinal tract and is an important aspect of the digestive process. Well, here's the expression of the receptor in the enteric nervous system, and you can see the brown coloration here. I hope you can see it, and the brown coloration in here is, these are the nerves in the Auerbach's plexus, and you can label them as well with class 3 alpha tubulin, showing the same kind of pattern present both in the longitudinal and the circular muscle in the bowel. This is colon, but again you can see this in the small bowel as well, and you can stain the same type of cells with nitric oxide synthase. So the receptor is in the enteric nervous system, both in Meissner's as well as Auerbach's plexus. So what do we think that this receptor is doing physiologically? So the receptor is not there to potentially be a target for treating diarrhea and evolution, but must have a role in the normal function of the gastrointestinal tract. We believe that there are a number of functions of the receptor, but I'm pointing really at just one today. So as we eat food, there's an important stimulation of a number of secretagogues that enhance fluid secretion and reduce fluid absorption. And these include substances like guanylin, which stimulate the release of cyclic GMP, and also vasointestinal peptide, which I've shown you, which will stimulate the release and generation of cyclic AMP. Both of these processes stimulate fluid secretion. Fluid secretion is an important part of the digestive process for mixing enzymes within the food and increasing the ability of the digestive process within the intestine. The food digestion then liberates substances, amino acids, for example, and polyamines, which are potent stimulators of the calcium sensing receptor. And I'll show you that this stimulation of the receptor does just the opposite effect of the secretagogues. It reduces fluid secretion and enhances fluid absorption. This, we believe, is a necessary part of increasing the absorptive process, increasing the ability to move these substances of digestion, nutrients, from the gut lumen into the body. And if you think about it, all the focus, or a major part of the focus in thinking about the gastrointestinal system has been related to this aspect of secretagogue stimulating fluid secretion. But there must be some mechanism for turning this off if you want to enhance the fluid and nutrient absorption. We believe that this is a major role played by the calcium sensing receptor. And again, I'll show you that indeed in experimental animals this is the case, and it provides a way to completely abrogate the fluid secretion aspects that come from secretagogues. So we know that bacterial toxins such as E. coli toxin STA, causing infantile diarrhea, traveler's diarrhea, cholera toxins, for example, from Vibrio cholera, causing the disease cholera, attack the system by activating the normal physiological processes. So bacterial toxins can stimulate secretion directly from the epithelial cells, as I said, but also can work through the enteric nervous system. Viruses can do both, but viruses, very importantly, at least best shown for rotavirus, stimulates fluid secretion by activating the enteric nervous system. And the receptor function in both of these areas, both at the epithelial side and at the enteric nervous side, is an important cause of diarrhea, and we'll talk about calcium receptor activating both of these processes and the potential to reverse this process. So if we look at the calcium sensing receptor, one of the first questions we asked is this receptor functional? And indeed, it is functional in the gastrointestinal tract. If you look at, and these are colonic crypts that have been isolated from rat. So this is an individual crypt. This is another individual crypt. And the crypts were loaded with fluo3, a calcium fluor, a calcium sensor, looking for a rise in intracellular calcium, because this is a major downstream signaling mechanism of the calcium sensing receptor, working through G-alpha Q, activating phospholipase C, generating IP3, and causing the release of intracellular stores of calcium and a rise in cytosolic calcium. And this is the same crypt after a very short period of seconds after activating the calcium sensing receptor with calcium. You can see a similar effect by activating with the polyamine spermine. So what I've shown here is log calcium. So this is the extracellular calcium outside the cells stimulating the calcium sensing receptor, and IP3 release. And calcium and the polyamines work together. The polyamine spermine is primarily an allosteric modifier. That is, it enhances the ability of the receptor to detect calcium. So you can see at an EC50 of around 1 millimolar for the gut, very similar to what we see in parathyroid calcium sensing receptor, for calcium in the absence of spermine. And if you add 100 micromolar spermine, you can see a leftward shift of the EC50 of the receptor. So adding spermine enhances the ability of the receptor to detect and respond to calcium, very similarly to a calciumetic. And this is the response to intracellular calcium, the rise in intracellular calcium. And you can see the same effect of spermine on shifting the EC50 for the release of calcium, again demonstrating that this is functioning like an allosteric modifier of the receptor, also demonstrating that this receptor can respond to agonists of the receptor, physiological nutrient agonists of the receptor. Now, many of the data that I'll show you today is from the isolated perfused crypt. I'll follow this up with some data in the small bowel as well. But I just wanted to tell you what that method is so that you understand what I'm going to show you in figures. So the individual crypts can be isolated and they can be perfused. The gut luminal side is put into a collecting pipette. The blind pouch side is punctured by a perfusion pipette and then held by an outer pipette. And you can perfuse this crypt. You can load a volume marker like tritiated inulin in the lumen, and by collecting the radioactivity over a given period of time, you can calculate the movement of fluid either in the absorptive process out of the crypt or in the secretory direction, fluid going into the crypt. There's complete isolation of the extracellular interstitial blood side of the crypt from the luminal side of the crypt, so you can look at very, very nicely net movement. Now, it was shown by John Geibel and colleagues a number of years ago that crypts both absorb and secrete fluid, as well as surface cells in the colon both secrete and absorb fluid all the time. So both processes are going on simultaneously. Whether or not there's net fluid movement into the crypt or net fluid movement out of the crypt depends on the balance of these two processes. Now, these two processes involve systems, and what I'm showing you is not the complex version of the transport processes. There are a number of other that are involved in this, but I've tried to simplify it a bit for today's discussion. So chloride entry into the crypt cell is by the sodium K2 chloride cotransporter NKCC1. The chloride enters the cell and the chloride is then secreted into the luminal side or the crypt space through the CFTR chloride channel, as well as potentially certain calcium activated chloride channels. So this is a predominant secretory pathway. It's activated by cyclic nucleotides, which activate both the entry step and the exit step. And these entry-exit steps can be activated by both cyclic AMP, cyclic nucleotides, and cyclic GMP, cyclic nucleotides. The absorptive process in the large bowel is through the ENaC sodium channel, the same sodium channel that's present in the kidney, as well as the sodium hydrogen exchanger. There's also a chloride bicarbonate exchanger in the apical membrane for chloride entry. So this allows sodium chloride to enter the cell, and then the sodium can be absorbed through the sodium potassium ATPase, and the chloride leaves probably through another chloride bicarbonate exchanger or other processes basolaterally. So these two processes lead to sodium and volume absorption. This process leads to secretion. If you put the crypt up and you just measure the basal net fluid movement, it's an absorptive movement. So the net movement of fluid is out of the crypt, it's absorptive. Now, if you block secretion, what happens to net movement is it goes up. And that's because in this basal condition, there is both absorption of fluid at this level and secretion of fluid at this level. And when you balance those two, you have a net volume movement that is like this. So it's important to remember that I'm going to show you net volume movement, but it includes both an absorptive flux and a secretory flux of fluid within the crypt. So this secretory process, as I've indicated, is stimulated by cyclic GMP and cyclic AMP through their cognate kinases, PKG, protein kinase G, or protein kinase A, which activate a number of mechanisms within the cell, both entry and exits for chloride, and stimulate the secretion of fluid. Now, the STA, the guanylin receptor, is the normal G protein coupled receptor, which leads to activation or accumulation of cyclic GMP and activation of PKG. And it's this receptor which is targeted by the E. coli toxins like the STA toxin. Cholera toxin doesn't attack a specific receptor, but it ADP ribosylates G-alpha-S, causing it to be activated continually. So once you have cholera toxin activating the G-alpha-S, it stays, it's fixed, it's there until the cell goes through its normal life cycle and is lost into the lumen and gotten rid of in feces. And that life cycle is between 5 and 7 days for the gut epithelium. So cholera toxin attacking this activating the adenine cyclase leads to a stimulation of the adenine cyclase that's fixed. And cyclic AMP increases dramatically in these cells, I think as we all know, and then activates the secretory process. So let's take a look using this model and activating the receptor. And I've shown you that extracellular calcium activates this receptor. And here's the crypt. It's put up in the basal condition, and here's the usual absorptive process that occurs. And then we've used a general cyclic AMP stimulator, forskolin, that many people use. This is 10 micromolar, but you get the same effect at 0.5 micromolar. So this is a larger concentration of forskolin, but that's okay. Now you shift from net absorption to net secretion. So this is diarrhea. And now if you raise the calcium from 0.1 millimolar, and this is free ionized calcium, to 2 millimolar, so you're causing a maximal activation of the receptor. In the continued presence of forskolin, so you're not doing anything with forskolin, it's still there, it's still stimulating adenine cyclase, and you get complete reversal of the secretory process, so you're back to net fluid absorption. Well, that's fine for forskolin, but really do you see this with a toxin? And the answer is yes. So if you look at cholera toxin, you see the same set of events. Crypt starting out with absorption, you add cholera toxin, and cholera toxin takes about 30 to 45 minutes on average for it to fully stimulate secretion, but you can stimulate secretion nonetheless at a level that's very similar to what you would see with forskolin. And of course, cholera toxin stimulates, like forskolin, the accumulation of cell cyclic AMP. And this is a typo. This should be cyclic AMP, not cyclic GMP, of course. And if you add 2 millimolar calcium, you can completely reverse the process in these crypts. What about the cyclic GMP-mediated processes? Well, the normal peptide agonist of the guanylin receptor is, of course, guanylin. That's secreted into the gut lumen from the pancreas. And you see the same process. So here's the net absorption. You add guanylin, you get now secretion. This is the diarrhea. And you can completely reverse this process by increasing gut luminal calcium. I'm not showing here, but you can reverse all of these processes either by adding calcium to the gut luminal side of the perfused crypt or the blood side. So the receptors are on both sides of the cell, and both receptors are, in our hands, appear to be equally effective. What about the toxin, the STA toxin, which attacks or attaches to the guanylin receptor? Well, again, you see net secretion, and you can reverse this process by increasing gut luminal calcium to quite high levels, but nonetheless you can reverse the process by activating the receptor. Now we looked at calciumetics because these calciumetics, these are the drugs that have been used to treat secondary hyperparathyroidism, very effective in activating the receptor through the superagonism effect, allosteric modification that I showed you earlier. And these are the curves for two enantiomers of this calciumetic R568. Now this is a quite potent calciumetic, and in fact, when Amgen was developing cinacalcet, the first agent that it looked at was this R568, and it went through phase 1 trials, very effective in lowering parathyroid hormone secretion. The only problem with the drug is that it was only metabolized by a single P450 enzyme in the liver, and there was some patient variability in blood levels. So this drug was abandoned for use in secondary hyperparathyroidism and was modified by medicinal chemistry to the compound cinacalcet that is now a commercial product. So this is a very potent calciumetic. And what's really intriguing about this is that the sensitivity of the receptor in the gut for R568 is in the picomolar range. You can compare this to the EC50 for PTH effect, and it's in a nanomolar range. So this is about 3 logs more sensitive in the gut to these compounds than in parathyroid. The R enantiomer is always more potent than the S enantiomer, and this is another indicator of action on the calcium sensing receptor. The drugs that are used, both cinacalcet and R568, are the R enantiomer, and the R enantiomer is the one that's most potent. Now we know that an important part of the secretion that we see in toxigenic diarrheas, cholera toxin, or E. coli STA toxin, are related to accumulation of their cyclic nucleotides. So if you take the CRIPS and you measure basal cyclic nucleotides and then you add cholera toxin, you see this dramatic increase in cyclic AMP accumulation in the cell. Many people have shown you this is what you expect to find. Now if you increase the medium concentration of calcium to 2 millimolar, so these are intact CRIPS, you see the complete reduction in cyclic AMP production despite the treatment with cholera toxin. So cholera toxin is there, it's ADP ribosylated, adenylate cyclase in these cells. It's generating cyclic AMP like crazy, but if you add calcium, you can completely reduce the accumulation of cyclic nucleotides. And this process is reversed by a phosphodiesterase inhibitor, IBMX. So in the presence of IBMX, you no longer are able to reduce the cyclic nucleotide generation. And we see the same profile with E. coli STA toxin, the same kind of event. You increase cyclic GMP, and you can reduce this down to basal levels by increasing calcium from 0.1 to 2 millimolar. And this process is completely eliminated. The effect of calcium is eliminated by adding phosphodiesterase, broad spectrum phosphodiesterase inhibitor, IBMX. Now what about the effect of this R56 compound, 568 compound? This is the same profile for cyclic AMP that's accumulated in the presence of cholera toxin. Here's the rise. And you can see that R568 in a dose-dependent fashion will completely abrogate the accumulation of cyclic AMP. And you see the same kind of profile with cyclic GMP. Guanylin stimulates it, you can get the same effect with STA, of course. Stimulation of cyclic GMP, and R568 will reduce this down to basal levels. So the effect of adding calcium, or the effect of adding a calciumetic, is to eliminate the accumulation of cyclic nucleotides, the central stimulator of the fluid secretion and the diarrhea. Now what does this mean for net volume absorption? So this we did with forskolin, but recent studies we've also shown with STA and with cholera toxin but showing you just for convenience the effect with forskolin, the stimulator of cyclic AMP. So if you have IBMX present at the same time that you add forskolin, of course, you see a very potent stimulation of secretion, which is what you would expect. But now if you raise calcium, you see no effect on net secretion because you've eliminated the accumulation, or the reduction, sorry, in cyclic nucleotides. And you see the same effect if you add R568 on top of the calcium. In other words, in the presence of a phosphodiesterase inhibitor, you completely lose the effectiveness of either calcium or calcium plus R568 to get rid of the diarrhea, demonstrating that an important aspect of reducing the fluid excretion by raising calcium or by a calciumetic is dependent upon a phosphodiesterase activity. Now, an important upstream component of this is a rise in calcium, and specifically the increase in the generation of inositol trisphosphate. So this is an inhibitor of phospholipase C, and so it completely eliminates the accumulation of inositol trisphosphate. I'm not showing you that here, but we've demonstrated that in colonic crypts. So what happens if you eliminate the first event in signal transduction by this receptor, that is the activation of phospholipase C? And you can see you completely lose the ability of R568, either in the lunar bath, to reverse the fluid secretion in the presence of this PLC inhibitor. Now, what about cyclic nucleotide accumulation? Well, this is the cyclic nucleotide accumulation that you see with cholera toxin. But if you do this in the presence of this PLC inhibitor, again, you completely eliminate the effect of R568 to reduce the cyclic nucleotide accumulation. So again, signaling through PLC generation of IP3 and increasing intracellular calcium are a critical component of the ability of the activated receptor to reduce toxigenic fluid secretion. And I think that from animal studies, the proof that this is working through the receptor is shown on this slide. This is a wild-type mouse, and this is the same kind of model that I've shown you previously, where you stimulate with either cholera toxin or guanylin, and you get this secretion, and you can reverse this with a calciumetic. If you eliminate the calcium-sensing receptor in this knockout mouse, then you completely lose the ability of R568 to eliminate the fluid secretion and the diarrhea. So an animal demonstration that what I've shown you so far with calcium, what I've shown you with calciumetic, is working through activation of the calcium-sensing receptor. Now, what do we think one of the mechanisms of action of this is? Well, we showed you that the activation of guanylin receptor by STA, the activation of adenylate cyclase by cholera toxin and generation of cyclic AMP, activation of their cognate kinases leads to fluid secretion. And what we believe we've shown is an important aspect of the mechanism is a calcium-dependent CAM kinase, we haven't shown you this, but CAM kinase-dependent activation of phosphodiesterases in colon epithelial cells that lead to the rapid degradation of the cyclic nucleotides. And this effect is so potent that you can reduce the cyclic nucleotides even in the situation where you fixed activated adenylate cyclase by cholera toxin. Really, we think quite a remarkable event and an extraordinarily potent activation of phosphodiesterases and metabolism of these cyclic nucleotides. Now, in the last couple of minutes, I just want to show you that it does more than that. So, not only does the calciumetics or calcium affect fluid secretion by nucleotides, but it also inhibits the co-transport activity, which is critically important for moving chloride into cells. And you can do this using colonic crypts and using a chloride-sensitive dye. So the important thing to see here is that if you have a cell that has a basal level of chloride and you add forskolin, what you see is an increase in chloride coming into the cell, and that's shown by a reduction in fluorescence of this dye. So this is chloride entry into the cell, and it occurs very rapidly when you use forskolin. If you add bumetanide and then add forskolin, you see no change. And so what about adding R56 and adding R568 in the presence of forskolin? It looks very, very similar to what you see with bumetanide. And in fact, you're eliminating chloride entry into these cells, and the chloride entry is specifically due to activation of this cotransporter. So R568 not only reduces cyclic nucleotides, but results in the inhibition of chloride entry. Again, a potent effector in reducing fluid secretion, chloride secretion and fluid secretion. Now, not only does the receptor reduce secretion, but it activates absorption, specifically in the colon through sodium-hydrogen exchanger activity. So I won't go through how we measured this, but it's looking at the ability of accumulated protons in the cell to be secreted out of the cell. And this is all sensitive to inhibitors of sodium-hydrogen exchange activity. So remember, this is a very important part of salt absorption in the colon. And when you activate the process with R568, you see a dramatic and significant stimulation of sodium-hydrogen antiporter activity. So at the time that you're reducing fluid secretion, you're also stimulating fluid absorption, and we think both processes are important in reducing the diarrhea. So again, not only reducing fluid secretion, but enhancing fluid absorption. So as I showed you in the beginning, bacterial toxins hijack the normal fluid secretion mechanism, and we're showing you that you can balance this effect by basically attacking and stimulating the anti-secretagogue effect of activating the calcium-sensing receptor by calcium agonists, which could be both nutrient agonists or drug calciumetic agonists. I wanted to show you just two other slides before I end, but to show you that this effect of calciumetics is also present in the small bowel, a very important part of the intestine that's involved in secretory diarrheas. And we can do this by mounting a sheet of the small bowel in a standard Wissing chamber, where you can measure short circuit current as an index of chloride secretion and therefore fluid secretion. So this is in the duodenum, and this is showing you representative traces, current traces, and what you can see is that if you add forskolin, you can see an increase in short circuit current. This is the fluid secretion that's occurring. And if you add the solvent for these drugs, you get no effect. If you add the calciumetics, you can see reversal of this forskolin-stimulated short-circuit current in the small bowel. We've also looked at this using ileal loops perfused in vitro. And so this is an ileal loop that's about 90 minutes after adding cholera toxin, and it's dramatically distended. It's like a balloon filled with fluid. This is another loop taken out of a rat in which we've added the same concentration of cholera toxin, but in this case 100 nanomolar R568, this calciumetic. And the volume in this ileal loop is the same as in control, and that's shown here. We can measure voltage across the epithelium, and I won't go through what this is actually measuring. But if you take an ileal loop and you add cholera toxin, you see a dramatic shift in voltage, and this is an indicator of chloride secretion and fluid secretion. And if you use the same concentration of cholera toxin, but now in the presence of R568, you can completely abrogate fluid secretion. I'm not showing you the data here, but if you now add R568 in this situation where you start out with only cholera toxin, add R568 at this point, at about 45 minutes, you can reverse the fluid secretion. So if you add R56 at the same time of cholera-toxin, or you add R568 after you begin to accumulate fluid, you can either stop or prevent the process or reverse the process. So what I've shown you this morning is that there are many, many different toxins, many different disease-causing factors. What we want to do is see if we can have a host-based therapy, which regardless of the type of vector, we may be able to stop the fluid secretion, because all of these vectors use a common set of pathways to stimulate fluid secretion. And we believe that the calcium sensing receptor provides a new target for potential therapy in these individuals. And with that, I want to thank you for your attention, and I'll be happy to answer any questions.
They are not closely related. You may consider answering one, but there is no sufficient
I can
time.
choose.
Okay. First, the steepness of the dose response curve to calcium suggests that there is a very strong positive comparativity with the Hill factor, much higher than 1. And I wonder if that has been studied.
Yes, so the Hill factor ranges between 3 and 5. This has been best studied for parathyroid, but it's also been studied in stably expressing human calcium receptor in CHO cells. You see the same thing, a Hill about 3 to 5. We think in part this is due to multimerization of the receptor. So we think there are multiple factors which are causing this very steepness of the curve with an EC50 of 1.2 millimolar calcium. That's really quite dramatic, that you have a low affinity receptor that can sense minute changes in ionized calcium. We don't understand all the processes that are involved in that, but certainly we believe that, and many other people do believe that, this multimerization of the receptor is an important aspect of this sensing mechanism.
My second question is the modulator that you have of activity seems to interact with the transmembrane domains of the G-protein-coupled receptors, and the degree of similarity of receptors in this region is very high.
Yes.
My question has to do with the selectivity of these agents.
Yes, certainly calciumetics at very high concentrations can activate other receptors or affect other receptors. I think right now the best example that we have is that this is working through the calcium receptor, is the calcium receptor knockout mouse. So if you completely eliminate this receptor, at least for the effects that I'm showing you on colon, fluid secretion, and absorption, you can completely eliminate those effects by knocking out the calcium sensing receptor. So we think that's the most potent data, important data, demonstrating that the effect that I'm showing you is most likely an effect on the calcium sensing receptor, and not effects on a variety of other receptors. We do know from work done at Amgen that the concentrations that we're using have very little effect on a panel of about 140 different receptors. So again, we think that at the concentrations that we're using, we're predominantly affecting the calcium sensing receptor.
And finally, regarding the pharmaceutical aspects of these super agonists or modulators, since there is a very strong effect on gastric acid secretion,
Yes.
I imagine that these compounds have to be covered in order to avoid the gastric action, or they have to be in combination with a proton pump blocker such as omeprazole.
No, I mean, the data from the therapy with cinacalcet in secondary hyperparathyroidism is that you don't need to coat them. They seem to be quite resistant to acid, and they're very, very rapidly absorbed. They're probably predominantly absorbed by the time they reach the end of the duodenum, or at least the first part of the jejunum.
I have a comment First of all, it's a wonderful job you have done in terms of the impact that this concept on blocking two different mechanisms of two prototypes of toxins to induce secretory diarrhea, which are very important in terms of public health in developing countries, in South Asia and many countries in Latin America. And for people like me who are being interested in finding new ways to intervene for preventing or treating these diseases. And it was something like people like Dick Garan, who work a lot on the cyclic AMP for the ST toxin. And we knew that calcium was very important in this process because we knew that the concentrations of the calcium influenced the production of cyclic AMP. But there were no really a good idea of whether we could block this process with chemicals like the ones that you have described that block the receptors for the production of for the concentration of calcium. And so it's so important to identify drugs that are affecting all the different cycles independently of the type of system. That I think it has a great potential. One of my questions is in terms of since this is a very broad effect in terms of calcium metabolism, there are many other tissues that are affected, particularly muscle. The effect in the contraction is quite dependent on the calcium concentrations, intracellular concentration of calcium. That could be an important side effect on the use of this substance. So could you comment on this?
Well, thank you for your comment. We're also very excited about the potential, and I must say it's a potential. It's not a proven therapy. It's a potential for therapy, but we're very excited about it because it does attack the host. It does attack the central mechanisms that lead to this disordered process that's in acute diarrheal illness. The best answer to your question is that the data that we have from the use of this calcimetic cinacalcet hydrochloride for secondary hyperparathyroidism, and in some patients have been treated for as long as 6 years now. There's no evidence of significant skeletal muscle effects. There's no clear evidence that the receptor is expressed in skeletal muscle. So I think there are certainly receptors present in a variety of cells in the body. Those could be affected by an absorbable calcimimetic. We're looking at non-absorbable calcimimetics as I sit here, as I stand here. So there are a variety of pharmaceutical approaches that one can take looking at is a non-absorbable effective, for example, in acute diarrhea. But I would argue that we're looking where the potential use of a calcimimetic is quite, in acute diarrheal illness is quite different than how we would use it in secondary hyperparathyroidism, where a patient is on the drug daily for essentially their remaining life. Here we're talking about an acute therapy, and a therapy that would involve the only a day or a few days use of calcium receptor agonist. I haven't presented to you the fact that the therapy that we're proposing and that we're currently working on to bring, we hope, over the next 3 to 5 years to phase 1 trials is both an augmented oral rehydration solution as well as a calciumetic. And the oral rehydration solution would contain things that you could imagine it contain: calcium, polyamines, amino acids, those kinds of things, other factors that we've identified, other nutrient factors that we've identified that can activate the receptor. So we see giving a calciumetic very short term, could be just a single dose or a couple of doses, to really stop the diarrhea and then maintain the individual on an oral rehydration solution containing both the sodium and the starch or glucose components, as well as activators of the calcium sensing receptor. I mean, for anybody who has been involved in looking at the effectiveness of oral rehydration solutions, which are effective, but there's a significant problem with them that every person in the field, every study has shown, and that is the acceptance rate of oral rehydration solution by mothers or caregivers of infants is very poor. And one of the reasons why it's very poor is that the traditional sodium starch mechanism in oral rehydration solution, which does nothing for secretion, it enhances absorption through sodium coupled glucose transport and volume movement, but it does nothing for secretion. So the mothers or the caregivers are giving oral rehydration solution because the physician or the person who delivered this packet to them said, take this and this will help, see no change in diarrhea. So diapers that they're changing and their infants and everything, so they're giving a therapy, say give this medicine, this will work, okay? And it does reduce mortality rate in acute diarrheal illness, but does nothing for fluid secretion. So the caregiver thinks, this isn't working in my child. The diarrhea volume remains the same. Okay? What's changing is that you're trying to combat this with increasing fluid absorption. So the acceptance rate has been as low as 10-15% in some studies. Rarely does it reach even as high as 70%. And again, I am told by workers from WHO and from others that the acceptance rate is a real problem. So having a therapy that not only enhances absorption but critically attacks secretion, I think is going to be very important for not only as a therapy but acceptance of the therapy by the mothers or caregivers. So you can have the best therapy in the world, and if the mother doesn't give it or the caregiver doesn't give it to the infant, it is worthless. And so stopping the fluid secretion, demonstrating that it has an effect on the number of diapers that they're changing, the volume of fluid loss, is going to be, I think, a real advantage in this kind of therapy.
Excellent talk, Steve. I just would like to ask you something about other cations that stimulate calcium receptor, like magnesium, vanadium, and some antibiotics. What about some antibiotics that stimulate calcium receptor?
Yes.
Are you comment on that?
So any of the polycationic antibiotics that have been looked at, prototype being gentamicin, activate the receptor, and they're actually quite potent activators. You can activate the receptor with polylysine, polyarginine, any polycationic protein or factor seems to be a potent activator. There are minerals that act not so much as activators but act as blockers. And these are metals that are generally considered to be toxic metals. And in fact, we've used activation of the calcium sensing receptor, which appears to block some of the toxic effect of these heavy metals. And we've used this in the marine industry, in the fish industry, fish farming industry, as a potential therapy to get around the toxic effect of heavy metals where there may be heavy metals in the grow-out areas for these marine species. And we're studying that interaction. It's going to be very complex, but from an operational standpoint, it appears that if you activate the calcium sensing receptor, you can block the effect of at least a part of the toxicity of heavy metals. Thank you, everyone, for your attention.